Having oxygen in your blood does not guarantee that the oxygen will reach your cells. In the lungs, oxygen enters the blood and binds to hemoglobin inside red blood cells. Each hemoglobin molecule can carry as many as 4 oxygen molecules. The circulation then transports this oxygen throughout the body. But oxygen still has to separate from hemoglobin before it can enter the tissue. As cells consume oxygen, tissue oxygen pressure falls, creating a gradient that favors oxygen movement out of the blood. Active tissues also produce more carbon dioxide as they metabolize fuel. That carbon dioxide diffuses from the tissue into the blood and red blood cells. Inside red blood cells, carbonic anhydrase rapidly combines carbon dioxide with water. This produces bicarbonate and hydrogen ions, lowering the local pH within the normal physiological range. Those hydrogen ions interact with hemoglobin and help stabilize a lower oxygen affinity configuration. Some carbon dioxide also binds directly to the globin portion of hemoglobin, further favoring oxygen release. Higher carbon dioxide, greater acidity, and increased temperature signal that this is a metabolically active tissue. Together, these conditions reduce hemoglobin's affinity for oxygen, allowing more oxygen to be released. This relationship between carbon dioxide, acidity, and oxygen binding is known as the Bohr Effect. On the oxyhemoglobin dissociation curve, the Bohr effect appears as a shift to the right. A right shift means that at the same oxygen pressure, hemoglobin holds less oxygen and releases more into the tissue. The released oxygen can now move down its pressure gradient, out of the blood and into the working cells. Inside the mitochondria, oxygen serves as the final electron acceptor, allowing electron flow and ATP production to continue. Carbon dioxide does not carry oxygen into the cells. It helps create the conditions that allow hemoglobin to release oxygen where metabolism is most active. That is the Bohr effect.